Next up, I'd like to introduce Corie Ralston, formerly at the ALS, but now on the bigger, better things with some cool, really cool new technology.
Thank you, Jay. Hi, everybody. I'm Corie Ralston. So I run the X-ray footprinting program here at the ALS. We're a very recent addition to this ALS-ENABLE resource. So we're somewhat of a new kid on the block here, but I'm really thrilled to be part of this awesome team. So I did want to start out with just a basic overview of X-ray footprinting because it's a lot of people have not heard of it and don't know what it is. So this is a very simple conceptual overview. So basically, it's a solution state method. We irradiate we have protein in some kind of buffer. We irradiate with X-rays. The X-rays interact with the water and produce hydroxyl radicals. And the hydroxyl radicals will go on to modify protein side chains. They're highly reactive with protein side chains, and to some extent the backbone. So we get these covalent modifications formed on the proteins, and we then digest the protein, usually with a protease, and we then run our resulting peptides out through LC-MS to figure out exactly where those modifications occurred on the side chain. So this is the same overview, but in a pictorial form. So basically, we produce our hydroxyl radicals. They modify the protein in certain places where they're accessible. We digest, and then we run an LC-MS. And let me just tell you briefly how you actually get information out of this method. So imagine you've got a protein in two different states. I got one on the left side of the screen and one on the right side of the screen. And let's say you know the structure of your protein in the one on the left there, the compact form. And you run this method, and let's say you see a modification, which I've indicated by a pink circle there on the screen. And then you run the same method, the footprinting method, on the protein in its other state. And then you find that there's these modifications that occur on the linker between the two domains. And so from this accessibility information, from the hydroxyl radical modifications, you can infer that the two domains have come apart. And so that's how you get from solvent accessibility information to structural information. And we can do this on a residue by residue level. So you get information about the accessibility of each residue to water, basically. So just a couple highlights through the years. This method can be applied to a lot of different protein systems and nucleic acid systems. So it's been used to look at gating mechanisms in membrane proteins. It's been used by Sarah Woodson's group out at Johns Hopkins. It's done some absolutely beautiful studies on looking at ribosome assembly actually in vivo, so in cells. People have used it to look at GPCR activation. How does those proteins interact? People have also used it to look at antibody antigen binding, so figuring out where epitopes are. So it has a wide variety of uses. It mostly gives you interaction information, where are proteins interacting as a function of time. So practical aspects of the method. So we generally use micromolar concentration of protein. We usually need about 500 microliter volume to get a complete data set. We, like I mentioned, we're generally comparing the protein in one state to another, and this is because different residues have different reactivity to hydroxyl radicals. So you want to normalize that out by looking at the protein, say, alone versus binding to another protein or binding to a ligand or changing its conformation. One sort of drawback, I think, to the method is that we're quite buffer limited because pretty much everything you can imagine scavenges hydroxyl radicals. So glycerol is a no-no, detergents will scavenge radicals. Generally, we're doing our experiments in phosphate buffer, which does not scavenge radicals. So there's that limitation. I wanted to let you know the exposure times on the order of microseconds, generally these days under just 100 microseconds or so. So that gives you a sense of how much kind of time resolution we can get if you're interested in some kind of kinetic study. We do have to use LC-MS as part of this method, and that is at the moment a bit of a bottleneck for us. We have access to LC-MS and we will do that for users, and that's no problem. But we don't have our own instrument at the moment, and we have a very long queue, and we have quite a few user groups now. So this method is not fast compared to crystallography or SACs. We're very slow at the moment. So I just wanted to let people know that. We do have an additional inline fluorescence measurement that we can do if you're interested. So This is our setup at the beamline. We have a syringe pump to deliver sample past the X-ray beam, and we can use a jet, or we can have sample flow through a capillary. We have a lens for looking at your sample and also collecting fluorescence from the sample if you're interested in collecting that at the same time as collecting your footprinting data. So that was a whirlwind introduction to footprinting. I hope people are interested in the method, and please feel free to contact us. So I have a very, very small team of people here. You can contact me or Sayan Gupta. And you also see in that picture Lena Christensen, who's a really amazing research associate in our group, and Brandon Russell, who's our engineer in the group.